Hello guys and welcome back to Excelsior Academy. So here I am with the second lecture for structural biology. So in the last lecture I just discussed like what is the plan in order to complete this particular playlist, and I'll be starting with the very basic today. Okay, so we'll just brush up our all understanding of the amino acids, proteins, and then we'll jump into the structural biology. So let me share my screen and let's start with the first lecture of this particular playlist series. Okay. So our first topic will be about like what is structural biology. Okay. So first thing we have to understand over here is that we are going to study structural biology in order to study the like where you want to apply this phenomena. For example, if you have the understanding or like the structure of a protein, okay. Let's say you are talking about a protein and let's talk about insulin. Insulin is a very good example for this. Now, this insulin, let's assume that we don't know the function of this particular protein. ठीक है तो अब क्या करेंगे like from the insulin you can do all your biochemical assays, okay, and from there you can understand what kind of reaction it is taking helping in taking place. Now what if you have the structure, now from the structure understanding its active sites or the binding sites, we can understand or we can predict actually the function of a protein. Okay, and that's why the important role of structural biology comes over here. It's not only to understand the function of a protein, but this will also help in drug designing. So eventually with the series and upcoming lectures, we'll be understanding and study how a particular drug design is, we can start off. Like if suppose you don't have the structure of a protein, then whether you can do a homology modeling on this and can predict the structure. And once you can predict the structure, you can also predict the function of that particular protein. Okay, now why it is important, as I mentioned, like this is going to help in your drug designing. It will also help in getting the drug inhibitor, enzyme action, enzyme activity, all those kinds of functions, whatever, in order to understand the molecular biology, it will help. Now, how do we do it? Very basic understanding is that first you actually purify your protein. Okay. Then once you have the protein purification, we go for the crystallization. I'm sorry, crystallization. Now, this can be done by X-ray diffraction. The other methods are like NMR. So here also you don't have to crystallize a protein, but you need a pure protein. And mind you guys, getting a pure protein is the very critical and the crucial step. And the other methods are like cryo-EM is there. So even that can be used in order to predict the structure. Then there are other software tools where, with the help of the bioinformatics understanding, you can actually predict the structure of a protein based on the available structures. So all these things we'll be covering in this particular lecture series. Okay, now moving on to the next. So now we understand like why this is important. Okay, okay. Now, what is structural biology? As I mentioned, so this is all this lecture notes is given over here. So structural biology, it is a scientific discipline that looks at the molecular structure of a biological macromolecules and how the structure is relating to its function. Now, why it is important? Because it will help in understanding how a particular molecule is working, what makes them the suitable for this particular task, and can we manipulate them to work even better or to do the other things. Now, here I will ask one simple question. This is really very interesting, and this was one of the question it was asked at the ISET Pune. Okay, so it was there for the one of the integrated PhD questions. It was, so your question is, we know that kinase, okay, kinase is there. What is the function of kinase? It adds your phosphate group, right? Now, to what functional group it adds? So if you know the function of kinase, wherever sorry. So wherever you have this OH functional group, it is going to add your phosphate group. It removes the H and adds the phosphate group, right? Now the question is water molecule, okay? So water molecule also have this OH group. Is it possible for your kinase enzyme to add phosphate group to a water molecule? This was the question. Now, Please try to get the answer and let me know through your comments. Okay. Now coming back to the topic. So as I mentioned, there are many other principles and the techniques like X-ray diffraction, NMR, cryo-EM, which we will be using in order to understand the structure of a protein. So when I say protein, it doesn't mean that only protein. It could be your nucleic acids, your carbohydrates, and the lipids molecule also. Okay. So NMR, XRD, and cryo-EM, all kinds of biological molecules can be we can reduce the structure of it. Please remember about this question and let me know your thoughts with the comments. Moving on to the next. Okay, now what do we mean by structure? So whenever we talk about the structure, what comes to your mind? Obviously the 3D and the 3D and the quaternary structure of a protein, right? Now let's talk about taking the example of a protein and let's go one by one. So when I say about the primary structure, okay, now what do you understand by the primary structure? Of course, it is made up of amino acids, right? Then what kind of amino acids are available? So we know that in the system we have got the 20 amino acids are there. Now, some can be small, which can be flexible. Some are hydrophobic, right? Then some are hydrophilic, where you can see the OH groups are there. Your basic and the positively charged amino acids are also there. We have got the bulky and the big amino acids like tryptophan, which like it is not possible for them to be present in the protein surface. Okay, so they will be like they will be buried inside of the protein structure. Now, so what is over here? Right? So we have got the different types of structure over here. One second, just let me remove these linings. Yeah. So what are the different types of amino acids over here? So we know that these are the positively charged arginine, histidine, and lysine, and negatively charged aspartic and the glutamic acid. Now, one small request over here. Now, if you really want to do very well in structural biology, it's a small request that you understand and remember the structures of all the amino acids. It's really very crucial because it helps you in understanding the electron density. Once you get the X-ray diffraction done and you generate an electron density map, it will help in getting the structure. Of course, there are computational methods by which you can put into the parameters the sequence of the protein and you can get it. But let's assume a situation where the electron density map is not fitting. Your sequence which you have provided, it is not falling into the electron density map. So there you need to have this imagination or the visualization of the amino acid structure so that you can actually reduce the structure. Okay. And just a small request. Sorry for all this. Then we have got the amino acids with the polar uncharged side chains, serine, threonine, asparagine, and glutamine. We have got the other amino acids over here like cysteine, cellulocystine, glycine, and the proline, and the hydrophobic side chains. This is really very important to understand and remember. Alanine, valine, isoleucine, leucine, methionine, phenylalanine, alanine, tyrosine, and tryptophan. These three are also your aromatic amino acids. Now coming to the next, the secondary structure. Now, before I proceed to the secondary structure, just again a basic understanding over here for everyone that we do have 20 amino acids. And how do we read those primary structure? We read it from your N terminal to C terminal, right? I know this is very basic, everyone must be knowing, but just for the sake of the basics, I'm taking this again. So N terminal we say because it has got your amino group, right? And C terminal because it has got the carboxyl group over there. So we read or it is read from N terminal to the C terminal. Okay, now we can move on to the next topic, that is our secondary structure. Okay, so now let's talk about the secondary structure. Now, when we talk about secondary structure, what comes to our mind? Anyone? So it's all about your alpha helices, right? Now, when you talk about alpha helices, then it should come to your mind that again it is hydrogen bonded and it has got 3.6 amino acids residues per turn. Okay, a quick revision notes. Then coming to the next is the beta pleated sheets. Okay, now for beta pleated sheets, what comes to your mind? Okay, there are two types are there. We have got the polar, we have got the anti-polar beta sheets. Then we have got the beta turn coil structures. These are also common and irregular secondary structures present. Okay, now let's see few examples over here for all of these alpha and the beta pleated sheets. Let's go to this example. Okay, so this is one example. Here we're talking about the alpha keratin, and here you can see that each filament, okay, each filament of your hair, like if it is a hair, so each filament it is made up of protofibrils, right? And each protofibril it has got the protofilament. And if you clearly see, see the protofilaments made up of, they are nothing but the heterodimer, and each heterodimer is made up of again only the alpha helices. Okay. Similarly, if we talk about the silk fibroin protein over here, you will clearly see that how nicely they are arranged. Okay. Now, anyone can tell me whether this is a pallor or the anti-pallor beta sheets? Go and check your basics and let me know through your comments. Okay. Now, next is like where you must have seen like hemoglobin structure is there, right? Now, hemoglobin structure is actually a tertiary structure. Sorry, it's a quaternary structure, but it is made up of all these subunits coming together, and all of these requires your both alpha and the beta sheets. We'll come to the hemoglobin later. Now, covering about the secondary structure, it is really important to understand that it really plays a very crucial role. Now, for example, there are many researchers who are working on the secondary structure determination. For example, you have got the FTIR. This is Fourier transform infrared spectroscopy is there. Okay, this also tells you all about your secondary structure elements. Then there is something called CD spectroscopy. You can refer to the techniques in the techniques or the methods in biology to understand the functions of these techniques over there. This is also part of your CSS syllabus. So CD spectroscopy, where you can actually get the percentage of your alpha, beta, or the random coil. So this will also give you information how your protein is made up of, like how much percentage alpha is there, how much beta is there. Or if you see that the percentage of random is more, so you can also talk about how uh, open your structure is, or like how close your structure is. So all these basic or the preliminary information you can get from the CD spectroscopy, which talks about your secondary structures. Okay. Now, so as we know, these proteins are all made up of 20 amino acids, right? So they can have multiple roles over here. Either they will be enhancing any chemical reactions, or they must be acting as a storage protein or the defense proteins. They must be involved in the movement of your cargo, protein cargoes, or they are involved in the cell communication, right? Now, anything happens at the secondary structure level, like any kind of misfolding happens, it can cause a disease. Now these can disease you must have heard is like Alzheimer disease hoga ya Parkinson hoga, Creutzfeldt-Jakob disease hoga. So all these also is happening because of the misfolding of the protein. So you understand that over here also the primary structure, sorry, the secondary structure is playing a crucial role. Now there are certain bioinformatics tools are there which can be used in order to predict, or you can study about the secondary structures. Okay, now these are the bioinformatics tool. So I would like to give over here in the information of for the secondary structure. Okay, so one is, please note down if you really want to explore that particular bioinformatics area. There is this thing called Sci Red. Then you have got the Potter. You can just open and you can browse through it, all this web page. This is freely available. Then we have got, then we have got Pro- Porter. Okay, I mentioned. Then we have got the Prof, and then we have got the Sable. Okay, so these are like different bioinformatics small tools are there where you can actually put your secondary structure, sorry, primary structure information. You can put it over there, and you can actually predict like what kind of secondary structure it is having. Okay, so that's all for your secondary structure. Let's move on to the next. That is our second tertiary structure. So here also, if you could see here, the example is given like one domain of the heme and the beta polypeptide chain is given. It is also having the alpha helices, right? Now the 3D structure. So it is determined by it is determined. It's a 3D shape which is determined by the interactions between all your amino acid side chains and the your hydrophobic interactions, right? Where you can have also the van der Waal interactions. So in the case of tertiary structure, we call it them as the domains. It's a tertiary structure of a protein which is made up of a one or more proteins coming together, and those are known as the domains over here. Okay. Now coming to this tertiary structure, these can be of two types. These can be of two types. Okay, one can be, it could be your globular and the other could be your elongated. Now, what comes to your mind? Elongated, or somewhere it is also known as fibrous. Okay, so globular, what kind of protein comes to your mind? So globular is like spherical something, right? So hemoglobin we can say. Now, when you talk about fibrous or elongated, collagen type of protein comes to the mind. So I guess so far so good. Let's move on to the next. Now we have got the quaternary structure. Now, about the quaternary structure, it is nothing but the arrangement of two or more polypeptide or domains coming together to form a complex protein. So, now it varies. Now, quaternary structure could be of different type of, the number of subunits. So it can have two or it can have four. Sometimes it can be eight, where, for example, in the case of the histones, you must have studied it is having the octamer, right? So eight subunits are coming together. In the case of hemoglobin, we have got the four subunits. Now, these subunits can be of homo, that is like same, identical, or this could be hetero, where you have got the different polypeptide chains coming together. For example, in the case of hemoglobin, now what do you think? Hemoglobin is what? Homo or hetero? Any guesses? So this all subunits are coming together. So in the case of hemoglobin, you have got two alpha chains are there and then two beta chains are there. Okay. Now they come together to form a quaternary structure for hemoglobin. Along with this, we have got the four heme groups. Okay. Now, what can disrupt the quaternary structure? It is very easily, you can disrupt the structure. Sorry. Either you can use basic heat treatment or you can use the chemical treatment. Okay. Minor heat treatment will also disrupt your quaternary association. It can actually becomes into the individual subunits. Now, if you increase the temperature, obviously the secondary structure can also be disrupted. But mind you, primary structure is quite stable and it won't be easily disrupted by the heat or the chemical treatments. Now, after understanding, having the basic understanding of all this thing, now we'll try to understand like how it is possible for X-ray and NMR to give the insight into the structure of this to go through it. So in our next lecture, let's talk about the importance of structural biology and the techniques used in the structural biology. So for today, only the basics were covered. Requesting everyone to go back and just learn your structures of all those 20 amino acid structure. This is really going to help you in further calculations and how the electrodensity map is plotted, how we are going to resolve it, also in understanding of the Ramachandran plot. So till then guys, take care and stay tuned to the Excelsior Academy. Thank you.